Thank you. I want you for a moment to imagine and think about what your life would be like without something as commonplace as antibiotics. How many surgeries that you've had could you have had? How many infections you've had in your life would have self-resolved? How many of us would even be here without the discovery of antibiotics? What I get really excited about is not just to think about the past, but what's happening now. So what revolutionary technologies could be as impactful as something like antibiotics? Well, I'm here today to talk about one of those, and it's the idea of hacking the immune system, and it very well could be the next greatest medical innovation since antibiotics. And I'll talk a little bit today about the tools needed to make that happen. So going back, I'm a big fan of history, if you couldn't tell. Go back 2,400 years to the time of Hippocrates. And those intellectuals and practitioners of medicine actually understood something really important, which is that our bodies are the true healers of disease. I mean, we treat disease now with a lot of different things, but if you just think about it, it's really something that happens within us. And what's going on there is we have this miraculous system called an immune system. And when I think about it, just think about how many things and pathogens your body has been exposed to, but that you've survived. And the immune system can fight those and battle those. And not only that, it actually has a memory. Your immune system remembers what it was exposed to. And that just blows my mind. As a scientist, as a businessman, I think that's incredibly exciting. And then if you think about something like cancer, some scientists speculate that cancer spontaneously forms in our body every day, cancerous cells. But it's our immune system that recognizes those and stops those. And that how we think about cancer, what we define cancer as, is really when the immune system doesn't recognize a cancer cell and it grows out of control. So the crazy, exciting thing about the immune system, and the question I'll pose today, is what if we could actually hack the immune system so that we could teach it or train it to recognize those otherwise unrecognizable cells, those cancer cells. To kind of get into that and explore that a little bit, we got to first take a look at what's in the blood. So there's over 200 cell types in your body. And if we kind of just zoom in on some important ones, don't worry, I won't ask you to remember all those today. I want you to remember one, which is a T lymphocyte, otherwise known as a T cell. And when I think about T cells, I don't know, for me, I like analogies. So think about a T cell as the tank of the immune system. It can shoot holes into cancer cells if it knows where to aim. But if it doesn't know where to aim, how can it go and deploy and kill the cancer? So there's a way to actually teach the immune system, something called cell therapy. A specific form of T cell therapy is called CAR T therapy. And I like to think about it as hacking the immune system. So to kind of look at that process, the first thing you have to do is you have to go and get billions of white blood cells out of the body. And then we got to get those T cells out, our tanks, out of all those billions of T cells. Once we have those, we can then activate those T cells, essentially turn on biological mechanisms so that it can essentially be genetically engineered, I like the word trained much better, to then be grown up or cultured so that you can put it back in the body, and now those T cells recognize the cancer that it didn't recognize before. And in doing so, can actually essentially completely eliminate the cancer from your body. So this is happening right now. There's 7 FDA-approved therapies for CAR T therapy, a lot of blood cancers. There's thousands of clinical trials, new clinical trials going on with this. Because just think about it. It's not just applicable to cancer. Maybe you could do this with viruses. Maybe you could do this with diabetes. And maybe even you could use this with stem cells to reverse the effects of aging. All those things are possible, and many of those are actually happening right now. There's work going into that. So it's an amazing technology. Just think about the future. What will a life after the broad deployment of cell therapy look like? And will it be similar like antibiotics, where the life expectancy was about 47 years before antibiotics? And think about what it is today. So there's another part to the story. Not all is well. As exciting as this is, not all is well. And the reason is cell therapy is incredibly complex. So you saw the process I showed. That can take as long as 3 weeks. It can cost as much as $300,000. And there's also massive limitations on how many of those therapies you can produce at a time, because it's basically customizing a patient, like single blood bag at a time, versus a lot of our drug molecules are manufactured in masses. So it's a very unique problem because we're using cells as medicine instead of compounds. And what that looks like in a real life setting, I wanted to share an example of that. James McGarry is a high school classmate of mine back in Bakersfield, California. James was a very healthy, strong young man, and he entered a battle with cancer. And on the right here, you can see where after chemotherapy, after radiation, when there was no other options, he entered a clinical trial for cell therapy, a T-cell therapy. And you see in this picture, that's where he's actually getting those billions of cells collected, those white blood cells, and then they would need to go do the T-cells and the process I showed before. But he actually passed away before the manufacture of that 3-week time period was complete. So we think three weeks is too long, and we can do better. To look in that in a little bit more detail, this idea of billions of cells, just to kind of give you the engineering problem, if I gave you a blood bag of mine and said, can you please get the T-cells? That's like giving you a bowl of 1 billion M&Ms and saying, can I have the blue ones, please? How would you do that? How would you actually deploy technology that can improve that workflow, that cell therapy workflow, and work in that engineering way to isolate those cells you want? So at Akadeum Life Sciences, that's exactly what we're doing. We actually use tiny floating particles. We call them microbubbles. And I'm showing here a video of a lab tube, and I'll zoom in here in a moment, where you'll see a blue floating particle. Those are the microbubbles. And we put antibodies on the surface of those so they can just attach like a biological lock and key to the T cells. After the microbubbles are attached to the T cells, they'll float like a little life preserver for the cell. It'll float those to the top, everything else sinks. It's the simplest way, in our opinion, you can do a separation for a workflow like cell therapy. The white in that bottle is an example of what this would look like if I had a big enough vial to share with the audience today. And that white layer, that's millions of microbubbles that have separated under gravity. The remarkable thing about this technology is how elegant it is, because it works in any size container, any shape, or any number. So you can completely change the workflow in very important applications like cell therapy. I wanted to show just an example of what we're doing right now here in Ann Arbor with applying that microbubble technology to doing those separations in blood bags. It's research stage now. We're moving toward clinical applications soon. But what you see here is a blood bag, those billions of white blood cells. And we can add microbubbles through one of the ports. And if you just hang up the bag, it'll basically separate on its own. You don't need anything external because you have gravity. And those microbubbles float to the top. That's what the white is. So there's hundreds of millions of T cells up there. All the other cells sink down. And then you can simply drain the bag, and now you've completed a separation. What's really important about this is how much more of the blood bags you can process at a time. Just with the separation where we're at today, we can do 4 times more at a single time. So what we see in the future with deploying technologies like ours and using microbubbles for both that separation step and the activation, that we can actually help solve some of the problems we see in cell therapy right now. We can have a faster vein-to-vein time because of the simplicity. We can reduce the cost and process more at a time, which means it's more accessible. Because think about those applications I mentioned before: viruses, aging, diabetes. Our insurance company is going to pay $300,000 a treatment? Probably not. So the job then we look at as our team is to make a contribution to making cell therapy more accessible. Our platform wasn't originally designed for cell therapy. We have many different technological applications and have actually implemented those. So things like testing wastewater for COVID. We have a partnership where we expect to see products out soon. And if you look at diagnostics, there's applications there, and even in fundamental biological research. So we're very excited because we believe in a future where cell therapy is more accessible to all. And we believe in a future where diagnostics are more meaningful and the results can be used more effectively. So with that, I'd like to say we believe the future is now, and that part of that is happening right here in Ann Arbor. So we'd ask that you join us for the journey. We have a QR code here and a link to our social medias. We'll have a table out on Liberty Street from 1 to 5. Thank you.